And without further ado, I'm delighted to welcome our very interesting PhD students that are doing work. We're showcasing work from the Weinberg College of Arts and Sciences, Kellogg, McCormick, and the School of Communications. So we're very happy to show representation across 4 different schools today. Our first speaker will be Melissa Manus from Anthropology, and she will talk about ecological and environmental determinants of the infant's microbiome. Welcome, Melissa.
Thank you. I'll go ahead and share my screen and get started. All right, does this look all right for everyone? Okay, so hi everyone, my name is Melissa Manus, and today I'll be walking you through some of my research on the ecological and environmental determinants of the infant microbiome. So when we talk about the human microbiome, we're talking about the collection of microorganisms and their genes that live in and on our bodies. And we know that the human microbiome mediates the effect of different aspects of our environment on our biology, physiology, and health. This occurs because our microbial communities are particularly sensitive and responsive to aspects of our environments and lifestyles, including the foods that we eat, the pharmaceuticals that we ingest, and the physical contact that we have with other people and other animals. These sorts of exposures directly influence the microbes that we harbor on our body, and these microbial communities in turn have myriad influences on different physiological systems and associated health outcomes. Now, my research focuses specifically on the infant skin microbiome for 2 reasons. First, the skin represents the true primary interface between the body and the surrounding environment, so it represents a good place to understand how different environmental exposures shape the microbes that we harbor. Now, additionally, human infancy is an exceptionally social period. So infants are born relatively sterile, meaning they don't really have their own true microbiomes yet, but upon birth and subsequent exposure to factors in their world, they're exposed to microbes and actually acquire these microbes, which establish into different communities across the body. So human infancy is this highly social period where infants are coming into direct physical contact with mothers and other caregivers. And this represents frequent opportunities for infants to actually acquire microbes from other people's bodies. So in my research on the infant skin, I apply an ecological perspective. And through this, we don't view the human body just as sort of a singular system with a singular microbiome, but instead the body as a meta-community of microbes, or this interconnected patchwork of different microbial communities that are serving as distinct ecological niches for microbes to live in. And so when we think about how the infant body is seeded with microbes, we can think about particular behaviors that promote microbial sharing on 2 different scales. So the first is microbial sharing between hosts, between host bodies. So for our example of human infancy, we can think about behaviors that help promote microbial sharing between mom's body and infant's body. But at the same time, when we think specifically about the skin, we can also think about how microbes might be shared across the skin sites of a given infant, right? So in this example, it could be that microbes on the infant hand make it to baby's mouth or other parts of their face. So I use this ecological perspective to broadly ask in my dissertation research how early life environments are shaping microbial communities across the infant body. And today I'll be presenting some brief results on a study that I just wrapped up, which recruited 52 families living in the Chicagoland area. For this study, I collected skin microbiome samples. So I took skin swabs from both infants and their caregivers. And this was coupled with extensive survey data on infants' physical and social environments. So the results I'm presenting today come from 16S rRNA bacterial gene sequencing. So this allows us to characterize which types of microbes are living on the infant body. And what I'm about to show you comes specifically from the infant skin swabs, not the adult swabs. So jumping right into some results. What you're looking at here are 9 different windows. Each window represents a particular type of microbe. So what you're looking at are the 9 most abundant or frequently detected microbes in this data set. For those who are interested, we're looking at these microbes at the taxonomic level of family. So what you're seeing here is within each window, on the y-axis, we're looking at the relative abundance of that particular microbe. And moving left to right along the x-axis, we're looking at that microbe's abundance in the infant armpit samples, then the cheek samples, and finally on the right, the hand samples. So I'm going to walk you through an example using one particular microbial family, which may be one that some of you are familiar with. So in this particular family, we have streptococcal microbes. And these are microbes that are found on the human body and are thought to be a core member of the breast milk microbiome. And so what we can see when we zoom into this particular microbe from this larger plot, we see that the relative abundance of this microbe varies between body sites, and we're seeing it in much higher abundances on the infant cheek and infant hand compared to the infant armpit. And so the question becomes, can we actually connect behavioral variation to these differences that we're detecting in infant skin microbial communities? And for this particular example, I believe the answer is yes. So what you're looking at here, we're still focusing on this one particular type of microbe. And now you're looking at its abundance on infant cheek samples only. So here, moving from left to right, we have different feeding modes. So we have exclusive breastfeeding. We have infants who have pumped and bottle-fed breast milk. And then we have infants who receive formula. And what we're seeing is that for infants who are exclusively breastfed, they have an elevated abundance of this particular microbe compared to their counterparts who are not. On the other end of the spectrum, infants who receive formula have a decreased abundance of this particular microbe compared to their counterparts. Perhaps this is unsurprising because, as I said earlier, we know this particular microbe is thought to be a core member of the human breast milk microbial community. And so perhaps it's unsurprising that breastfeeding promotes babies' exposure to this type of microbe. However, I think what's really interesting to think about with the infant cheek in particular, we can think about the act of breastfeeding and how it's very likely to put infant's cheek in direct physical contact, not only with the skin on mom's chest, but also breast milk itself, right? You can imagine that baby's face is right there on mom's breast while baby is feeding, and breast milk is very likely to spill out, get on baby's face. And it could be that as breast milk is coming into contact with baby's skin, those microbes actually have an opportunity to settle and colonize baby's face. In contrast, we can look at the abundance of this particular microbe on baby's hand samples. So here you're looking at the same 3 panels with the different feeding modes, but with these hand samples, we are not detecting variation in the abundance of this microbe based on feeding method. And so in contrast to the cheek, which again is likely coming in direct frequent contact with mom's body and her breast milk, it could be the case that hands don't have that same pattern of contact and so are not receiving that same microbial exposure. And to bring this home even further, here the same panels now looking just at the infant armpit samples. So here, not only do we see really no variation in the abundance of this microbe by feeding type, but it's also barely detectable in these armpit samples at all. So to summarize, I hope I've given you a sense of the utility of an ecological perspective, as it helps us to think about how microbial communities may vary across the infant body. And it also guides future research to actually focus on specific behaviors that can work to promote microbial dispersal to the infant body and across different skin sites. So with that, I'll say thank you for your attention. Thank you to my funding sources, and a huge thank you to the people listed here. First and foremost, my study participants, but also a very special thank you to all of the people in our lab who helped with this project. I received a tremendous amount of help, ranging from actually collecting the samples to the large amount of lab work that it takes to do something like this. So thank you all very much, and I look forward to taking questions after we hear from the other speakers.
Thanks so much, Melissa. That was really very interesting. Next up, I would like to, and please put your questions in the Q&A now, just in case before you forget towards the end. Next up, I'd like to introduce David Sabin-Miller with McCormick's Engineering Sciences and Applied Mathematics. He will be discussing randomness inside nonlinear dynamics. Welcome, David. I think you need to unmute, David.
There we go. Hi. Yes, we good? Hello. So there's a bit of a shifting gears as we go to the engineering department. First of all, can people hear me now?
Yes.
Okay. So I'm David Sabin-Miller with the Applied Math department. And so I'm going to be talking about a recent bit of work we've been doing on a new kind of random, how to deal with a new kind of randomness in the world. So do a little bit of background because we're a very diverse group of individuals. A lot of things in the real world have, they evolve in continuous time, but they have like spikiness to them. They have randomness and jump around a bit when we look at their behavior over time. So sometimes this is just because of things we don't know. It appears to be random, or it's being influenced by something else that's changing really quickly. And so we get the spikiness. And so the first canonical example is Brownian motion, where you have a particle traveling through a fluid and it's being bumped by trillions and trillions of other particles every second. And so all these little bumps add up on the particle and creates this kind of randomness, no matter how fine of a time scale we look at, down to, like nanoseconds. It looks, it still has this many bounces in any given time period. And so it's effectively random when we simulate them, that we want to simulate a bit of time, and we add some randomness from all these kicks that we can't know about. And these kind of models are used all over the place, from finance, like the stock market, ecological models, neurological models. Anytime we have kind of messy systems where we can't know all of the many variables. And so you see, it could come up quite a lot in complex systems. And one note before we go further, we're talking about a continuous time evolution. So if something is happening once a minute and you roll some dice each minute, you can just add that, and that's, there's no problem with that when it's nonlinear. But I'm talking about, can we do this continuous time randomness with a nonlinearity, which means complication. We'll get into that in a second. So standard, there's a kind of informal way of writing down how we think something might be influenced. So this is a differential equation. We have how something changes, x is our thing, is related to, there's some part we call the deterministic part, which is, like the bulk flow of the liquid, for instance. There's no randomness to this part. And then we have part that controls how random the bump is over time. And so this eta, looks like a curvy n, is representing the random element. And this was Einstein talking about Brownian motion, first of all. And it took a little bit to get it so that it made sense, because if we think about classic randomness, if you roll a die 1,000 times and add them all up, it's basically like you're adding 3.5 every time. Like, things tend to converge to their average effect, but not in the case of trillions and trillions of little particles hitting things. So like, we get this randomness that exists over time evolution, and we can't use just regular random variables like rolling a die to represent that. So it was figured out by two different ways how to make this consistent mathematically so that we can simulate this. And it looks a little bit different, but you see mostly the same. We just have a deterministic part, and then this dw essentially acts as the randomness. So this is where you're rolling a die essentially when you simulate, but you have to scale it differently. I won't get into too much of the specifics here, but there is plenty of theory once we get to this Ito land for how to deal with these things consistently. But we notice that this format is kind of limiting. Eta here is just multiplied by a scaling factor, and then that whole thing is just added. So this is more restricted than you might think, as we'll see in a second. There are other ways where this eta might come up in theory, especially when we're talking about modeling complex systems where this can't really capture it. So this is an example of where it works great. We have this will provide an attraction towards 0. And this will be some diffusion bump, this random bump. And that would become this system. So that should look pretty clear that this dw is being the randomness. But here's where we ran into a problem. What if the rapid fluctuating quantity, the random quantity, has some nonlinear effect? It's not just added, scaled and added. So like this thing, if x, our quantity, it becomes fuzzy, and then the nonlinear cubing operation is added. How does that change? Like, how can we make sense of that? Because the former theory no longer works. And so the reason that I got into this is because I wanted to create a model for how the environment influences individuals' political ideology, that we're influenced by lots of different things. But some of those things are attractive and some of those things are repulsive. There's this, so this is a cubic function that I cooked up. And I wanted to simulate people experiencing a lot of different things over time. So rather than privilege some particular time step that we're updating every minute, I wanted to say, well, let's just say it's continuous, and that's an approximation for many, many influences. But then I wanted this input to be random, and there's no theory that exists for how to reliably simulate that. So it's just a slide from a previous talk. Don't worry too much about all this, but this is just the idea. It is a cubic equation, slightly different than the one we're going to be talking about, but this is the core essence mathematically of what we need to be able to solve. So here's what we propose. We take this, in this case, there's a random thing centered at x. So maybe we have x equals 1, and we have this normal distribution around it of eta. And then when we cube that and negate it, we get this weird looking distribution, because all of these things near 0 get sucked even farther into 0, and that's why we get this spike. And the big half of this gets cubed out, and it's much wider than the half that's near 0. So we get this weird distribution, and here I've just marked where the mean is, because that's one of the things we might want to preserve in the behavior of how we expect this to behave. Like, if this is a distribution and we cube all of these things, we should at least have the mean behavior be preserved. And basically, when we add up these many times, only the mean and the variance per time, kind of like the standard deviation, if these were regular randomness, matter. And so essentially, this is going to turn into the equivalent Gaussian distribution after this transformation. So it'll get a lot wider in this case. And once we're back with the Gaussian, we can do that old Ito formulation. And so it looks a little more complicated, but that's what this system would become under this calculation. This guy determines the mean, and this guy determines the variance per time. And so this is sort of our proposal of what this should mean, because there is no existing theory to deal with these kinds of situations. And I wanted to make models that could have, a more general appearance of unknown quantities. And so in general, we can now have this format, which is more general than the F and G, little f, little g format we saw before. And we're saying that, and we solve in our paper for what this looks like for a class of these systems, that what we should simulate is this ethos system where we preserve the average behavior and the variance over time. All right, and I think that's about my time. So feel free to leave questions in the chat. And
right on the money. Thanks so much, David. Appreciate it. And yes, do add your questions in the chat and we'll address those at the end of the talks. Next up, we will hear from Dawei Wang, who is a PhD student in management and organizations at Kellogg. His discussion will be on animal prints and how the loss of privacy continues. Welcome, Dave. Dawei. Mute. You're on mute there.
Hi, everyone.
There we go.
I'm a PhD candidate at Kellogg. And today I changed my topic slightly. I wanted to present a work that is forthcoming at Journal of Personality and Social Psychology. It's also on privacy. So yeah, let's get started. Because I understand there's a diverse audience here, so I want to give a very brief introduction to the topic. So in 2018, there was an article published in the same journal, in Journal of Personality and Social Psychology. So the article, the authors found that algorithms can predict sexual orientation better than humans. By the way, Journal of Personality and Social Psychology is the top peer-reviewed journal in psychology. And what the authors found was that algorithms can predict really at really high accuracy rate of sexual orientation. And they found that when you give the AI or the machine learning model 5 images for men, it almost perfectly classified the person's sexual orientation. So what does this mean? Why can algorithms predict sexual orientation? So the authors conducted some analysis to unpack the algorithm, and they found that it is mostly due to biological differences. So if you see on the left-hand side images, these are the images that the authors formed using the many images in the data set. So they averaged the facial images to form these images that you see on the left. So they found that heterosexual people's faces are very different from gay or homosexual faces. And they concluded that it is mostly due to biology. They said that the same gender sexual orientation might have stemmed from underexposure of male fetuses and overexposure of female fetuses to prenatal androgens responsible for sexual differentiation. So they concluded that because people's faces look different according to sexual orientation, that this must be a very dangerous situation for privacy, because if people upload their faces onto their social media, and because their faces are distinguishable according to sexual orientation, then algorithms can be used to detect people's sensitive traits, such as sexual orientation. So the paper generated a lot of media attention, was covered by a lot of famous media outlets, and then it was also highly cited. But there are a lot of questions that remain unanswered. So one of the questions was raised by this researcher from Chicago Booth. So they conducted a survey of yes/no questions. So they asked people yes/no questions such as, do you wear glasses? And they found that across all age groups, there is a significant difference between how heterosexual people and homosexual people, they report portraying themselves. So homosexual or gay people, they report that on average, they are more likely to wear glasses compared to heterosexual people. So this basically created a question of, oh, is it because of facial, structural, or biological differences, or is it because of how we want to portray ourselves? So, and you might ask, there might be question, okay, is it because of short-sightedness, or is genetic differences in short-sightedness? So they found, they also asked questions such as whether you're more likely to wear contact lenses or not. So they also found a difference. So basically, there is this complementary or explanation of what is happening, and I wanted to test this complementary explanation. So complementary being people also chose to upload certain photographs that portray them, that represent themselves. So I began to collect my own data. I used a public dating website, and I crawled basically all the profiles in the dating website using 4 fake profiles. And by the way, everything is approved by the Northwestern's IRB. So I collected a total of about 15,000 images. I'm sorry, 15,000 users, altogether 30,000 images. And I balanced the images by the class, by their age, and by the number of images. So you see here, the numbers are all matched between different classes. So in the first study, I asked the question: do presentational styles vary by sexual orientation? And you can see here on this graph, on the x-axis is the differences in the mean, and for each of the on the y-axis you have the different presentational styles. So the differences mean the differences are, the attributes are also measured in probability. So the differences in the probability will mean that, for example, glasses, females, homosexual females are on average 8% more likely to wear glasses compared to a straight female. And then in terms of smiling, you see here, straight females are on average 10% more likely to display a smile on their facial images compared to lesbian women or homosexual women. So that's, and this result is replicated in men as well. And the differences are, and the attributes, the differences in attributes are almost similar across the different genders. So men, straight men are 6% more likely to display a smile compared to their counterpart, to the gay counterparts. So in study 1B, I asked the question of whether sexual orientation is predictable. What is the predictability of sexual orientation using these presentational styles? So I conducted a cross-validation, a 20-fold cross-validation, and trained the model and conducted a 20-fold cross-validation. And the results you see here are the average cross-validated results. And we can see that using these facial attributes, we can predict sexual orientation at 0.61 AUC score. So which is higher than random chance. And remember that I'm only using 12 presentational attributes. So these are only just 12 numbers. and they can already predict at a 0.61 AUC score. This is slightly lower for men, but it's still above random chance. So next up, I want to use the images itself. So I want to use images, the entire facial images to predict sexual orientation. And first, I want to replicate the model, the sexual orientation prediction model in the previous author's work. So I use this model called VGG Face. It's a deep neural network. It's a pre-trained, I need to say that it's a pre-trained deep neural network, so everybody can access it. And it is a facial recognition algorithm. It's not trained on sexual orientation. I merely use the algorithm to extract some scores representing each facial image, and then use that score, input that score into a logistic regression model to get the sexual orientation output. And we can see here that using this very simple deep neural network pipeline, I can predict sexual orientation at close to 0.674 AUC score, which is much higher than random chance, and it roughly replicated Wang and Kosinski's results. So with the model, I want to test even further what's happening. So Wang and Kosinski said, in their article, they mentioned that the deep neural network was able to ignore the background of the image. So here, I want to test whether the deep neural network really ignored the background. So to do that, I basically mask the images progressively until at the end, right before the random. The random here is where I input the image completely as black image. So everything is black. So right before random, I have 96% covered. It's not shown here because due to space constraint, but 96% of the image was covered. So only 3% at the very corner of the image is remain, and it was given to the deep neural network model to test to predict the sexual orientation. So we can see that when the image is almost entirely covered up, only 3% of the pixels at the border are remaining, we can still predict sexual orientation at close to 0.55 AOC score. And for men, this is higher, and we see that the degradation in AOC score is not is not high as much as female. It's actually very small compared. And notice here that only 3% of the pixels are given. So next, I want to test the brightness, how the brightness of the image is different across sexual orientation. So, so what I did was I created 2 sets of masks. So to test whether the background, to test the brightness of the background, I masked the face. To test the brightness of the face, I masked the background. And I did this because I wanted to explore further what does the 3% of the pixel mean, right? There are only 3% of the pixels. What do they mean? And I was thinking they might be brightness of the image. So here by masking, we see that here first, the plot on the y-axis, I'm sorry, on the left, on the x-axis, we see the background and the facial region. So these are the brightness by different regions. And on the y-axis, we have the brightness. And we can see here that brightness varied according to sexual orientation, but and as well as facial region. So facial regions are on average brighter than the background. And in terms of sexual orientation, we can see that straight women are on average, have on average brighter facial image compared to gay or lesbian women. For men, straight men have darker facial image compared to gay men. And this is this variation. It's similar according to by background and facial image. So if background is brighter, your facial region is also brighter. So basically what this means is that there is an overall brightness difference in the facial image. And whether I wanted to ask whether this image brightness can predict sexual orientation. And I did this by blurring the images progressively until only 1 pixel is left in the image. So the 1 pixel basically is the overall brightness of the image. And I found that even 1 pixel is enough to predict sexual orientation above random chance. And this is this effect is much stronger for men than for women. All right. So what can we conclude from all these? So I want to just give a very brief theoretical implication and show this very famous sociologist's work here, and it's called presentation in everyday life. So this means that people, when they upload their photographs, they don't randomly upload. They choose the kind of photographs to represent themselves. So the deep neural network is actually picking up on these behavioral differences. And in terms of privacy and policy implications, people cannot protect themselves by wearing masks. So unless we are fine uploading photos that you might think might not look good, in other words, you just randomly upload your photographs. Unless you do that, you need to think about where you upload your photos and what kind of photos you upload. And with that, thank you.
Thanks very much, Dawei. Very interesting research and ties nicely, I think, into our next speaker's discussion, who will continue the privacy conversation. I'd like to introduce Priyanka Nanayakkara, who will be joining us from, did I get that right? Okay, good. Who will be talking to us about visualizing privacy, utility trade-offs in differentially private data releases. Thanks for joining us.
Thanks a lot. All right, so I'll be presenting this joint work on visualizing privacy utility trade-offs in differentially private data releases. So organizations like hospitals oftentimes have large amounts of data that might be useful for the public in some way. So for instance, hospitals have patient records and they might want to release an aggregate statistic about the rate of hypertension in a certain patient cohort. But releasing this aggregate statistic also puts there's some confidentiality risk. So an attacker might use that aggregate stat to not only find out what the rate of hypertension is, but also who specifically was diagnosed with the condition. So to prevent against this, practitioners can use differentially private mechanisms. So these mechanisms produce statistics in a way that preserves privacy. But using these mechanisms is tricky because it requires setting a privacy budget, also called epsilon. And I'll get into the reasons why this is like a hard task. So for these reasons, we decided to build an interactive visualization interface that helps a practitioner use these differential privacy mechanisms, specifically by helping them set privacy budgets. So before I get into the interface, I'll talk a little bit about what differential privacy even is. Here's the formal definition. It's kind of a lot to take in, so I'll just say at a high level, if an analysis is conducted under differential privacy, then we expect that whether or not any given individual's data is included in the analysis, we should output roughly similar results. And so in this way, differential privacy affords some notion of like individual level privacy. And also I just want to point out here that in the definition is that privacy budget parameter epsilon once again. And so that's going to end up being a knob that we can tune to control the strength of privacy protections. And I'll describe that more in a bit. So how do we actually achieve differential privacy? Mechanisms that achieve DP often work by adding a calibrated amount of statistical noise to results in such a way that then protects privacy. So importantly, epsilon is what controls how much noise is added, and in turn, the strength of privacy protections. So how do we actually go about choosing epsilon is a context-dependent policy question that requires considering multiple probabilistic trade-offs. And that's what makes this such a hard task. So for instance, if we want very privacy-preserving results, then we might decrease that privacy budget. That's going to increase the amount of noise that we're going to add to the results, which of course decreases the expected accuracy of what we can make public, but that also increases the strength of privacy protection. So that was a lot that I just said, and it's also a lot for anyone to keep in their head when they're trying to consider these privacy budget values. And so that was a lot of the motivation for this work. How do we simplify some of these trade-offs? So with that in mind, we came up with these 4 design goals for the interface. So we wanted the interface to first help a user understand the trade-off between epsilon and accuracy. Second, the trade-off between epsilon and strength of privacy protections. And third, statistics are oftentimes used to make inferences about a population. And so we wanted to also help a user understand how confidence intervals under differential privacy are impacted and how these different sources of noise propagate. And last, we wanted the interface to help a user take a total privacy budget and then split it among some number of queries and decide how much to allocate to one versus another. Right, so our interface is called Visualizing Privacy, or VIP for short. And this diagram just shows a bit of an overview of how VIP sits within a larger framework for sharing data or calculating these statistics. So mainly the data curator is the one who's going to be interacting with VIP, and they're going to provide a query. So maybe they're interested in a rate of hypertension, and they're also going to provide a privacy budget that they're interested in experimenting with. VIP then accesses a confidential data source, so that might be the hospital's databases, and then it visualizes to the data curator this risk accuracy trade-off. And the data curator can then iteratively choose different values for the privacy budget and see what the implications are to accuracy and risk. And finally, once they come to a conclusion, they can decide this is an appropriate value for epsilon for my use case. And then VIP can produce a statistic that can be made public. And that goes to the data consumer. In that case, that might be members of the public who are maybe reading some publication online. Okay, so now I'll get into a walkthrough of the interface. This is what it looks like overall. This interface currently has demo data that's around rates of hypertension for these different subgroups. So for instance, rate of hypertension by different ethnicity subgroups, so Hispanic or Latino, and not Hispanic or Latino by different age groups, different race categories, and then different zip codes as well. And I've also put a yell key down at the bottom where you can actually go and play with this interface. So if while I'm talking, you want to play with it, please feel free to go to that link, and I'll leave the link up for a bit. Okay, so let's step through each of the portions of this interface. The main point of interaction that a user has with the interface is through the Privacy Use tab. And so this tab has a privacy budget slider that goes with each of the queries. So for instance, at the top, we can see E corresponds with the ethnicity query. And so here, the user can change that privacy budget slider to play with different values that we're going to allocate of the privacy budget to that query. And then they'll see how these different results like accuracy and risk then update. To the left, we have an accuracy panel that corresponds with each query. And so here we're basically trying to show what is the expected accuracy of a privacy preserving release if you are to use the privacy budget that you've set on the slider. So the vertical black lines show basically whatever's in the database. That's no differential privacy noise added. But then the red lines, those animate, and those show potential privacy preserving releases under a given privacy budget. The dot plots below show a distribution from which the privacy preserving releases will be drawn. And this is kind of interesting. So when you move that privacy budget slider, the quantile dot plots update accordingly to show you how that distribution changes. In addition, the hypothetical outcome plots, so those animated red lines, also update their animations accordingly. So I also mentioned that the interface helps with statistical inference. And so what you see is the gray bars, those stay static, and those are traditional confidence intervals that we might construct in a typical setting where we don't have any privacy concerns. Above those gray bars, there are sets of colored bars, basically show what confidence intervals would look like under differential privacy. At the bottom right, we show a disclosure risk curve, and this is a way of representing what the privacy implications are for a given privacy budget. So here, each dot corresponds to each one of the queries. And so changing that privacy budget slider again shows how the disclosure risk that corresponds with that query would change. To evaluate the interface, we conducted a user study with 16 participants. Everyone had experience analyzing private or sensitive data, but nobody had any differential privacy expertise. So we asked participants to complete a set of tasks using both a spreadsheet that served as our control and VIP. So the spreadsheet did allow people to input different values of epsilon, and then it would return error values for privacy preserving releases, for example. We found that VIP helped participants do significantly better on CDF judgment questions and confidence interval comparison questions. So for CDF judgment, that meant that we asked questions like, what is the probability that the privacy preserving release will fall within this range? And for the confidence interval comparison questions, that was things like, how many times wider do we expect the privacy preserving confidence interval to be compared to the traditional confidence interval? And we also found some evidence that VIP may help with the budget splitting task. So that's where we gave participants a total budget, and then we gave them some accuracy requirements and risk requirements and asked them to split the budget across queries while satisfying all those requirements. So based on our study, we have some evidence to believe that VIP may help practitioners understand differential privacy, specifically by helping them keep track of these important relationships between epsilon, the privacy budget, accuracy, and privacy. And it may also help them make quick calculations that are relevant for setting privacy budgets, like calculating the CDF of a DP output distribution. As future work, we'd like to incorporate context-appropriate information into the interface. So that might be things like on the privacy budget slider, actually writing, in the past, someone at your organization for a similar data release used this value for the privacy budget, and to see sort of how that impacts privacy budget decisions. And we also hope to create normative frameworks for evaluating DP interfaces like ours. Okay, with that, I'll end. I just want to say thank you so much for having me, and I'm really excited to hear all your comments and any feedback on this work. I can be reached online, also in the Q&A, and if you're interested in the full paper, it's available at this Yauki. Thanks.
Super, super. Thank you so much, Priyanka, and thank you to all of our speakers today, Melissa, David, and Dawei. We do have a couple of few more minutes for some questions. One has come in from Luis Amaral. I believe this is for you, Priyanka. We'll go backwards, given that you ended the talk. Let's start you with the questions. Did I understand correctly that larger privacy budget corresponds to greater risk of disclosure?
Yes, and that's one of the counterintuitive parts of differential privacy. And that's, if you play with the interface a little bit, I think it becomes a lot clearer. But basically, smaller privacy budgets mean that we add more noise and we're protecting more privacy.
And was that, just a follow-up on that, I'm wondering, it's such an interesting tool, and thanks for allowing the team to play around with it, or the community to play around with it. Is that something you have considered looking into patenting at all?
Oh, no, I haven't.
Okay, just curious, just curious. Dawei, maybe we will go to the question posed for you. I saw earlier you were perhaps answering the question, but maybe would you like to answer that question live? The question was around the danger of potentially the research that you're doing.
Yes, sure. So, yeah, definitely, it is very dangerous for governments to use these technologies to predict people's sensitive traits. But as I typed in my response to Adam's question, it is also related to how often and how likely people are to express themselves online. So how likely people leave digital traces online. So so the more people leave their digital traces, the more likely you are able to predict people's sensitive traits. So I guess Yeah, so I guess there are basically two sort of components to privacy, revealing people's privacy. One is whether you have the technology, and one is whether people upload their photos or upload their leave digital traces. Yeah.
And I think your research, Dawei, is less focused on really what people might do with this. You're just trying to understand what can be done, looking at what people post online, as opposed to the applications or the potential use of that, if I'm understanding correctly.
Yes, yes, and I think this also speaks a lot to the policy world as well. So I'm trying to create more discussion for a better policy formulation. So right now I'm on the job. I just finished the job market, and seems like a lot of places that have policy implications, like George Washington, or I also got a job interview at Carnegie Mellon, their information system and policy school. So seems like they are all very interested in connecting my research to the policy formation realm. So I think my research is also trying to contribute to that world. Yeah.
That's great. That's great. And I think, Adam, we did just unmute you. If you had any follow-up questions, feel free to chime in, or I can move on and ask David a question.
Just
David.
a quick
Sure.
follow-up to that. So I think that's really fascinating, and how it's not just about the facial features, but how they present themselves. And I'm curious, is there any evidence that people post these pictures kind of out of demand? Let's say, are homosexual men more attracted to a picture that is brighter? So that people are presenting these types of photos because that is found more appealing, or does it just happen to be the kind of photos that they're posting? Is there any evidence for that?
Right. So I guess your question is mainly going into whether they are consciously or subconsciously or deliberately or indeliberately portraying themselves in a certain way. So in my research, I guess I can't really answer directly to that. So, but I'm just looking at in terms of privacy, right? What are some of the behavioral features that can be extracted from a certain digital trace and use that to predict certain sensitive information? But that is certainly a very interesting question, whether, yeah, so different people, they portray themselves so that they want others to like them better. So I think there are also a lot of very traditional past
Thank you.
I'm not sure.
Yeah.
I think we lost, or maybe it was just me. I lost your voice there for a couple of minutes, Dawei. But thank you. I didn't mean to jump in and cut you off, but given the time, maybe we will move on with just a couple of more questions, if that's okay with you. David, very interesting mathematical conversation or presentation you gave to us. I'm wondering if you might comment on the applications of this.
Yeah, so it's mostly in opening up so you can make models of nonlinear random things, but you have to privilege when it updates. You have to choose some time period. And often that's unrealistic to, you can impose something, but sort of the only way to be consistent with any reality is to go all the way to continuous time. And so that this is essentially letting us simulate these more complicated systems, which, like real world, my example was political ideology evolving over time. That it lets us simulate those at the continuous time limit, where there was no way to do that without it blowing up or becoming totally deterministic before. So it's pretty exciting that it could, if people see it, if it catches on, it could widen the scope of models that we can apply to the world in this sense, where we don't have to put our modeling bias, our choice of how it updates in there.
Right.
It can be continuous.
Very fundamental discovery, which is really exciting. Thank you. Melissa, we had a question also from Luis. I'm not sure if you had a chance to see it in the chat. It was about breast milk being sterile. Could you comment on that, please?
Yeah, it's a great question. Yes, so breast milk is not sterile. For a long time, we thought and assumed that it was. I think in part because for many decades, we assumed that all bacteria were bad, right? And we now know that's not the case. So in addition to breast milk delivering things like antibodies, we also know it's delivering specific microbes to baby's body. That being said, the vast majority of the research has been on breastfeeding and how it shapes the infant gut microbiome, just given the logical connection between what baby is consuming and eating and how it would shape the community in the gut. But when we think about the other part of the question was how microbes would be getting to breast milk. And so this is where, again, sort of thinking about different body sites ecologically becomes important. So there's sort of two potential ways in which microbes are getting into breast milk. I think they can be complementary. So kind of very quickly, one is that microbes could be traveling from other parts of mom's body to the mammary gland and getting into the breast milk during that particular period of motherhood. It could also be the case, and probably is the case, that as baby is feeding, they are sending microbes back to mom's body and could also be getting into the breast milk. So when we think about when babies are suckling, there's sort of this backflow that's happening. And so microbes from baby's mouth, or in my opinion, other parts of their body, like potentially their cheek or other skin sites, could be contributing microbes into that breast milk. And so you sort of have this sort of a backwash mixing scenario where you have microbes from all of these different sources and all these different body sites combining into the milk itself.
Right. Thank you, Melissa. And thank you, everybody. This is just such a delight to see so much diversity and exciting work that's going on with our PhD students. So the Lightning Talks really are just so exciting. And thank you all for sharing your time and your research with us. This has been a really great overview of some of the great work going on here at Northwestern. And I'd like to invite everybody to please join us again next week when we will have Fabiana Zolo joining us from Venice, and she will continue with a conversation on misinformation. And we welcome you there. So everyone, we're giving you all a great round of applause, and thank you so much for sharing some of your wintry day with us today. Thank you.